Welcome to session 8, Drug Discovery Part 2. I have the pleasure to share this session with my colleague, Professor Khaled Hussein. Today in this session, we have four speakers. It's our honor and pleasure to have with us today Professor Mohsen Mazar. Professor Mohsen Mazar is a professor of pharmacology and the Dean of Pharmacy in the British University in Egypt. In addition, he was the Dean of Pharmacy at Turquoise in Canadian University, at Canadian University, and very famous scientist in the field of pharmacology. Today, he is going to lecture us about drug repositioning and novel drug delivery system. So please.
Thank you very much, Dr. Ashraf, Dr. Khaled. Good evening to all of you. I would like first to thank the organizing committee for inviting me and my friends from the BOE to attend this conference for the third year. This is not, it's only the first time to actually deliver a lecture. I'm also glad that beside giving time for clinical pharmacy in a good conference like this related to breast cancer and a lot of clinicians attending, this year they really made some time for a very important two sessions, the drug discovery. For us as pharmacists, I think this is really very disappointing that Egypt, until now, for all years of experience and a lot of experts in the field of pharmacy as scientists, we have only maybe one or two drugs, which we call Egyptian drugs. So today I'm trying to elaborate more about why Egypt does not have contribution to have drugs. So don't expect this talk to be very scientific. It's more of a concept talk. To make things more clear, we should go through these points. The process of drug discovery is really lengthy and very expensive, and I'm sure in the last talk you know all about this. And because of that, only international pharmaceutical companies have the ability to discover and develop new drugs. International pharma compounds are mainly driven by market need, means they are profit-oriented, which is really understandable. Egyptian pharmaceutical companies are more concerned by developing generics. They have to wait for years and years until the patent expires, and then all of them goes for icy peanuts. This does not give much profit, but for them, because they don't have the resources for expenses and things, they go for the easy stuff. Academic researchers in Egypt who have a lot of experts in all fields of pharmacy, but due to lack of finances, financial resources, and interest of national companies, of course, they really have difficulties to go from molecules, as we've seen in the last session, to very important talks about new molecules who have new targets, which can be drugs, but would those can go through from Egypt as new drugs for treatment? Actually, we can see those as Egyptian drugs? I don't think so. And I know two eminent professors here of pharmaceutical chemistry will agree with me about the difficulties to go through. Not only the resources and the interests of national companies, but also of legislations. I'm sure you patent your compounds. These are new, promising compounds. Patent is very expensive. Is it only in Egypt or worldwide or in America? This is a lengthy procedure as well. And more is the legislation of the health regulatory agencies here, where it's not an easy to register a new compound. I know that. I mean, many committees in the health and what you call the Dar Saydala, and they don't have room for or special legislation if you have a new compound for registration and laws and everything. So the main aim of the present talk is to highlight alternative approaches to speed up the process and, of course, minimize cost, hoping that could be this is the way for Egyptian companies and Egyptian scientists to have new drugs. We're all familiar with this, what I call funnel-like. The traditional way for many, many years to discover new drugs is by having many molecules can sum up to about, you need about 10,000 or more to end up by only one drug. And this is the lengthy procedure, takes about 15 years. And to develop one in the traditional way, it takes needs about $1 billion. So, can we really skip some of these processes to make new drug, or have new drugs with really less expensive and in a shorter period? This one just, a breakup of the expenses as percentages, and every stage or every phase of these can really consume too much money and needs a long time, and I may go back to this slide again in a different way. So what's the alternative? And should be economically feasible approaches which can be adopted by Egypt at least. We'll look about Egypt and similar poor countries. We should concentrate about those drugs which are already known. They had FDA approval, and as you can see, we have about 10,000 drugs already tested for certain indications, for certain applications. All drugs we use, not including the generics. We're talking about the main drugs. This can come about 9,000 if we take off combination of drugs or different salts or the biological molecules. If we can screen FDA approved drugs for another activities, for another indications, can that lead to new compounds? Can that be patent? Can that be of interest to our companies to go through it in a less cheaper way and faster way? The approach for discovery is to find new uses for existing approved drugs. This is termed drug repositioning or drug repurposing, and traditionally has occurred by serendipity. Serendipity means it came by chance. Some cases, few cases. I think the one I should mention now is the discovery of sildenafil to treat erectile dysfunction. It was not designed to treat that condition, but by chance, not only chance, but good observation of clinicians that they found a certain effect happening. It was designed for heart disease and something, and someone by chance, with good observation, found that, and then you cannot rely on that to discover new drugs. You cannot just wait for chances. The main thing to apply this approach, we need and make use of the huge information we have at the moment. Huge information about the drugs, their effects and their side effects and so on. And it had to be in a library type, things that when you can search for a drug, you can easily find it. And as I'll show you later, how to use those libraries to test or predict a molecule which is already known as a drug, which can have another effects as an indication. So this is the drug bank. All chemists know that. You can go through it and really search for known drugs. If you needed the FDA-approved drugs, you can find it there. But this is another important database, which is called Zinc-12 and Zinc-15, where it can search for molecules. We have information about huge number of molecules which are not even processed to be drugs. It's even, as you can see there, by the information we have from these molecules, even if it is failed to go through other phases of clinical trials, but the information we have from their activity, pharmacological activity, beta activity, side effects, these are stored in databases where we can retrieve or at least simulate if a new compound will have a similar effect or not, so you can predict by these databases what you want to do. I already gone there, or you just gone back? Okay. So, I'll highlight what's meant by drug repositioning and drug repurposing, especially by using computational methods. And you've seen in the last session how that were really actively used in silico testing and high throughput screening as well. We should talk about drug combination as well. Drug combination is, combination of drugs which are already approved drugs for certain purposes, for synergy, and I should add here combination also with the purpose of having less toxic effect. If you add a known drug to another drug which is really used for certain indication, but you experience certain toxicity, so you can decrease that by a combination as well. Synergy can be patented here in Egypt and everywhere. So the new combination, even if two known drugs, you can patent it. And this was found successful in cancer chemotherapy, or cancer chemotherapy based on regimens with combination of things. And recently in Egypt, you can see that how, for treatment of HCV was also used. I also like to talk about new drug delivery system, how can formulate known drugs, approved drugs in new formulation, especially to target certain diseases, improve what's in there in the market, and how come that can save time and money. If you break up the money and time for the major phases of drug discovery, this you can end up with about 15 years, sometimes less, maybe 1 billion, sometimes less. But if you have a new drug, a drug known for certain activities, FDA approved to be used by humans, and then you discovered a new indication, you really will pass all of these main three parts. You save several years and a lot of money because the drug is already known, the drug is already tested, the drug is already approved. If you're going to use it in human by the same dose, same regimen, there will be no problem. It needs only approval for new indications and then can go to the market easily. This is really experienced by clinicians who always know about off-label indications. Drugs being used for certain indications, and by experience he can use it for another indication. But you need to have evidence for that, to approve it by FDA. If you have a drug, when you formulate it in another form, pharmaceutical form, especially if targeting, and you're going to use it in the same concentration or same dose, you can bypass all of those time and have really in short time to go there. And it can be patent as well. Patent really very important for pharmaceutical companies. Pharmaceutical company would not go to buy a drug from you unless it is patented. They need it to preserve their rights at least for 10 years before others can do it. So patent is really an important thing to preserve your right. If you have a drug combination, two drugs together, maybe you will have to go back there. Drug combination, even you can search for certain interactions, so on, but if you're going to put them together, treat a patient together, you may need to go back for some clinical trials, limited clinical trials, to make sure they are still effective and still safe in that case. And those are really the regimen to establish basis, evidence for these applications. So you can save a lot of what I just mentioned. I'm not going through details about computational method. I think in the last session you've seen two excellent, more actually, the four lectures were really excellent, and they were based on how they can retrieve a protein from the protein database or data bank and look for ligand abilities, binding abilities, and this technique is really important, not only for academic but for industry as well. They use that, and that can save a lot of time in the first section, especially if we're looking for new molecules. Now we're looking for the molecule existing drugs, but looking for other targets. So we can sit there. Still needs, I have to stress, still need the expertise and skills of pharmaceutical chemists to show. It's not a game. Not everyone can go sit there and try to play and see which one binds. It had to be purposed in a certain way that the skills of the pharmaceutical chemist will tell you, oh, for that we should go. It has that functional group or that group there, and then we can look for the other things. What's important, I think that was a question being raised to the last session. Would that will be very predictive? Yes, it can be predictive, with a high percentage as well, because several studies have established relationship between molecular ligand molecular modeling or structures and biological activities, which means if you have the protein structure and you show that this molecule, a new molecule or an existing drug, bind to that molecule, so you can predict, this is a new molecule, you can predict with certain 90% or more that it will have an effect. But of course, it will need to go through more. As I showed you in the last slide, computational modeling, in silico testing, and the high throughput screen, these all complementary tests. You can just stop with the molecular modeling. If you couple it with in silico testing and in vitro, at least you'll have higher predictability of the effect you're looking for. Several examples there, and we've seen several examples with FDA drugs, approved drugs for found new transporter inhibitors in vitro, which represent attempts in understanding off-target effect. Off-target effect means the effect which not mainly intended to. Examples is numerous. I don't have to go through this. If you wanted to search more, I think you can go to this a review article with Shao and Zinketer. Maybe I'll show you a snapshot of the actual reference. I can tell you more about this. This again about drug bank and docking And you've seen examples of that, and I might show you more examples. I'm just skipping things which is really. I didn't finish 20 minutes.
Yeah, sorry.
Did I finish 20 minutes?
Yes.
My goodness. I'm not going to go through all of that. You have minutes. It's actually, I have to tell you about thalidomide. This is a typical repositioning. Thalidomide, you know all the story of thalidomide. It was one of the drugs with the highly safety profile and sedative, and most important was an antiemetic. So it was recommended to be used during pregnancy as antiemetic because of high safety. And after a while, they found this phocomelia, and the drug was already withdrawn from the market in 1957 or something. After a while, they repositioned the drug as an anti-cancer, especially for multiple melanoma, and you can see this is the old drug which is dismissed, and then we have a new drug now. They didn't have to go through all of those clinical trials because the whole profile is known about it. Okay. I might mention him, robot scientist. It's called Eve, where you can go through a molecule quickly for molecular modeling and in silico testing as well. And this is the reference. Again, I should really talk here. That's okay. Something wrong. This is the end of it, huh? You want me to finish? Companies, to make companies, international pharmaceutical companies, they have to look for profit. They have the money to spend, it's an investment, they spend more, they will gain more. But if you go for two types of disease, what we call neglected diseases, tropical neglected diseases, or neglected tropical diseases, and orphan diseases, you have two situations where companies, international companies, would not invest huge money for that. Even though for neglected tropical diseases, there is millions who are going to use it. But those millions are in countries, poor countries. If you invest a lot of money, the new product will be highly expensive. And those companies would not buy it, even if you have many patients. Orphan diseases is the opposite. Orphan diseases is diseases where few sufferers. So if you invest money and have very expensive drug, too less people are going to use it, and of course you do not with a profit. So you'll go with this type of models for especially those. And for even pharmaceutical companies will be interested as well in this type of repositioning to find drugs for those at least two types. So we have types, it's called HIV predictor, where you can put the molecule, the name of the drug, even if it is an approved drug, and it will tell you it will have a sort of HIV or not. The same was very successful for treating malaria, treating schistosomiasis, treating trypanosomes, all those neglected diseases. Maybe from our results quickly, I'm not going to go through it, but the papers are all published. We found that valproic acid is really having the ability to bind histone deacetylase, and the histone deacetylase inhibitors some of them are known as an anti-cancer. So you have an anticonvulsant drug which should affinity to the molecular structure there of HADAC. Another thing which is phytanic acid, which can bind to these heterodimer receptors with bioglitazone or rosiglitazone in a way that can by itself has an antidiabetic activity, and in combination with rosiglitazone at least that have an additive effect, so you tend to decrease the dose of rosi. Obticolic acid, this is one of our recent publications. Obticolic acid is mainly an approved drug for primary biliary cirrhosis. It shows an anti-cancer efficacy against HCC in that. These are in vitro studies and mechanistic effect as well. And the most important about that is why it is the suggestion of the proposal was due to agonistic activity on a certain important receptors called FXR. And to prove that this anti-cancer activity is related to that, we used an antagonist. And by using antagonist, the IC50 is really increased here, which proves that, just in a minute, you blocked it there, it seems. I want to show that's only one slide of the new drug delivery systems. This is another slide for finding activity of an antidiabetic drugs by blocking to another type of adenosine receptors. You heard about this in silico. In silico approach, there was a question in the last at the last session as well. Would in silico approaches can detect adverse effects or not? It can, and it was tested. So we can really predict the side effects. So we can at least minimize the effort to do a lot of toxicity, so it takes time, animals, and so on. For novel drug delivery system, as I told you, you can use it to have controlled delivery, to make ideal drugs, and particularly for targeting, and several models who are interested in pharmaceutics, you can put it in micelles, and liposomes, neosomes, thiosomes, or even in nanocapsules. Many forms. This is our recent publication as well of using methyl dihydrojasminate. This is the one component of the oil of jasmine, and it found in vitro to have a good IC50. When you put it in a formulation, maybe this is against breast cancer. So we used here MCF7 as one cell line, but what's important to go for in vivo. And I think this is a good example. Probably the formulation here has less IC50 than the actual drug or the pure compound, but when you go there for in vivo, and in vivo this is the early cyst carcinoma induced tumor, subcutaneous or intramuscular, and by treat application, actually this is transient. Targeting here is by local application, dermal application. So for breast cancer it can be used. Instead of having systemic application, you can apply it outside, and you can see the formulation have a good inhibition of the tumor. And again, when you compare it with the void capsule or the standard, still much more important to produce effect. You can have here, this is another, actually this is only yesterday where we had the proof for that paper. If you have an extract of pomegranate, of the peel of pomegranate, this is known to have an anti-cancer activity. But the problem was all plant extract that it is highly polar. If you take it orally, it would not go through the systemic circulation, would not produce an effect. And that's where we can really target it by using certain formulations like solid lipid nanoparticles, and the particle there is very small, it's about 50 nanometers. And you go through several optimization process to find the proper formula or the form, and then of course we have to test it. And you can see that it is very effective, mostly against the MCF7 as an anti-cancer, and in vivo it's really there effective as well. And I think that goes to that. Thank you very much.
Thank you very much, Professor Mazar, for this interesting presentation. And I hope that you may use your influence in pharmacy administration to let them adopt repurposing or using drugs in combinations as like Egyptian medications as well. Okay?
For the delay, maybe we shouldn't have stopped Dr. Mazar, that's why. So I would like again to thank my professors here for sharing that session that I'm going to give you a short talk. Actually, I'm not going to take the whole 20 minutes. Share with you a quick talk about cytotoxicity through kinase, arsenal inhibition, small molecule inhibitors design and systems. So as my colleagues here, my professor and my colleague, Dr. Khalid and our young colleague Samar has already highlighted that kinases are an important set of proteins in the cell that can be targeted for management of many types of diseases. And why is that? As I have mentioned before, that the first step in the drug discovery process is drug target identification and validation. So why do you think that human kinome is a valid drug target? First of all, the human kinome makes up to 20% of the human genome, which is 1/5 what we have in our genes, encode for kinases. Over 500 known protein kinases have been discovered so far, and they can be classified into membrane and cytoplasmic type of kinases. The membrane ones are known as receptor, and the cytoplasmic ones are known as non-receptor, but they all share the same function, which is putting a phosphate group on a protein. And where on a protein and a specific amino acid, as someone has noted, either a tyrosine or threonine and serine, and to a lesser extent on an histidine. As you can see here, this is the phylogenetic tree of the kinases we know so far, but who knows, maybe there is more hidden in a pocket somewhere or there and will be discovered in the future. I'll go back here to tell you that the kinases comprise 8 families. They are subdivided into 8 families. So what they are there for? 500 proteins in one cell. What are they doing there? So they are responsible for a lot of activities, including signal transduction, doing a cascade of protein phosphorylations. Multiple signaling pathways are there. They relate information from outside the cell to inside the cell in the cytoplasm, and from the cytoplasm finally to the nucleus, where eventually a whole lot of processes take place. So one of the cellular responses that are important are those related to proliferation and cell survival, which is highly, highly connected to cancer cell survival. Other than that, we can see activities related to metabolism, secretory processes, and many other inflammatory responses, and many other types of responses. So, in that sense, we should say that protein tyrosine kinases are good guys here. They shouldn't be killed, right? But the thing is, they should, sometimes. So they have a good face and a bad face. The good face sometimes they act as tumor suppressors, and to our surprise, yeah, they do. But in specific types of cancers, they can suppress somehow brain cancer. Also, they can suppress lung cancer, and they can suppress. They can be pro-apoptotic in some incidences and helping chemotherapy. Yet on the other hand, sometimes they change their mind and turn into the bad guy, where they can act as tumor promoters in that case, either through overexpression or through mutations, where they become irresponsive to stop signaling. So whenever we find them that they change their attitude, we need to have an action which is stopping their action, either through small molecules, which is an easier way, and many drugs have found their way into the market through that way, or through monoclonal antibodies. So here this diagram shows the regulation of tumorigenic programming by activation of the kinases. So they play a lot of role in keeping cancerous crazy cells alive and functioning and dividing and metastatic. So the whole hurt that's made with the cancer can be mediated by the set of kinases. Well, the trends and achievements in development of small molecular kinase inhibitors has been long established. The first kinase that was discovered early in the 1950s, and that was a SERC family kinase. And after that, after target validation and identification and validation, the discovery process started. So the first drug that came into the market, as has been mentioned before, is imatinib, and actually it's a tyrosine kinase inhibitor. So came after that a few other tyrosine kinase inhibitors, plus serine threonine kinase inhibitors, plus dual protein kinase inhibitors, plus lipid kinase inhibitors. So you see that the market is still there. Yet if we see that through the development phase since the 2001, which is I think the first drug introduced, to the 2015, FDA has approved 28, I think now there are 30 kinase inhibitors only. So 17 years for 30 kinases, this is not a very fast-paced drug discovery process. But if you see the interplayers here, the big pharma companies that are trying to take a share of the cake, you find that big names are there. Pfizer is one of the highest players, share market holders. GlaxoSmithKline, then come Novartis, AstraZeneca, Bayer, Boehringer Ingelheim, and then others. So, do you share with me that we can add to that ocean? Oh yes, I would say yes. Usually because when we want to share in a project, we see the feasibility, and we have very great chemists that can teach an arsenal, not of kinase, arsenal of students to do good chemistry with a drug design concept and trying to target the kinases that we have. Also, there are yet unexplored areas now that every day, as we talk, X-ray crystal structures are coming out of new, yet unexploited, undiscovered, unused kinases. So we have the new kinases, and we have the chemists and the minds of the chemists and the tool of the molecular modeling, as Dr. Mazar and our professors, distinguished professors here, have highlighted. This is the progress of action again. So the first drug to come out was in 2001, and the process is still ongoing. And if you see what I meant by that, there are kinases yet unexplored. This is a family, maybe 2-3 families of kinases that have not been used as drug targets, and the crowd is all up there. We are just covering 1 or 2 families, and actually a few members of these as well. Those are the structures. So as the passion always comes, again, chemist loves the benzene ring, and this relationship will be ongoing till the end of the days. So we look for the structures. What are they? Because from the structures we know the structure-activity relationship, and this is the key to switching on or off a kinase. So yeah, the FDA has done a good job on that, but we as chemists, we have to explore what are the key structural elements which we call pharmacophores, so that we can utilize them and build upon them in the future. As you can see here, maybe you can see, I don't know, the data shows not that great, yet there are small, simple molecules that can be done in our labs with ease. No high-tech, no fancy thing. When we think about the pharmacophores, we think about the chemistry aspect. It's doable, it's tangible, it's easy to make, but we have again to keep, as I have highlighted in my first talk, the structural plus the physical chemical properties in our minds. Okay. So a lot has been done. You will find too many reviews talking about what traits of physical chemical properties should be in a molecule to be able to bind to a kinase. Among these are molecular weight aspects. So you find that most of them lie in the 500 range of molecular weight. And the number of rotatable bonds, which is really, really important because with the number of rotatable bonds, when the drug goes to the active site, it has to lose some of its entropy to be able to bind to the target. The number of hydrogen bond donors, acceptors, and the number of rings. This is a survey of the 28 or 30 now compounds that are out there. Mechanism, functionality, selectivity, therapeutic indications for these in the market, as have been highlighted kindly by one of our speakers, I think Summer did that for us, and Dr. Khalid as well. He highlighted that there are too many types of kinase inhibitors. They can be either type 1, 2, 3, 4, up to 6 now. And the main game goes mostly through around the type 1 and 2 kinase inhibitors. Less about the 3 to 6 inhibitors. So what is the difference? To us as medicinal chemistry, we ask, okay, scratch our heads now. What do I need to do to make it a type 1, not a type 2, or a type 2, not type 3, and so on and so forth. So structural biology has solved the problem for us and answered the question. So the difference between type 1 and type 2 is they both bind to the active site of the ATP active binding site. And the only difference is DFG in, DFG out. If you know what that is, it's according to kinase common terminology. Here the DFG is in, which is in the activation loop, oops, and here it is pointing out that small amino acid. It's only one small amino acid that can make a lot of difference. What about type 3? Type 3 are those that are allosteric inhibitors, but they bind to an allosteric site that is close by the ATP binding site. Type 4 are allosteric inhibitors too, but they bind to a distant allosteric inhibitor, allosteric site away from the ATP binding site. So when we look at the closer look, which has been already covered, it's typical for kinases to have a conserved kinase structure. So they have an N loop, and the N loop is mostly beta sheets, and the C loop, which is mostly alpha helices. Between the N loop and the C loop is what we call a hinge region, and there is what we call an activation loop. Where the ATP binds usually is in the hinge region, and the activation loop is what keeps things under control. It is either perhaps closed or open at active site. And if we look at how ATP binds, then we find that there are important hydrogen bonds by virtue of the adenine moiety here, some by the phosphate through a magnesium ion that is present in the active site, and the ribose plays a role. So again, it will be safe to work on the type 1, type 2 inhibitors. So I would now share with you quickly some of our findings against kinase inhibitors. The first class was fluorochromones. Those were not explored before as kinase inhibitors, they were just explored for so many reasons, but we decided why not try them with kinases as anti-cancer agents. So we have made a big series of compounds over here. I'm not going to show all the details of the results to be quick, But the in vitro cytotoxicity of these compounds, I'm showing the most potent, was done against them. So a panel of 60 cell lines. I'm showing the most prominent out of them. So we find that they are active in micromolar range with 1 digit, 2s, 1s. So they show that they are potent fluorochromones. Those compounds were coming from coumarin, which is known to be safe with a good safety margin. We took them further to try to understand how they really work as anti-cancer agents. So we put them on cells and tried to find what about the GSK3 beta level, which is a kinase, glycogen synthase kinase 3 beta. And there is a very well-known glycogen synthase kinase 3 beta inhibitor called flavoperidol, which is used as an anti-cancer in clinical trials. And we also tested cyclin-dependent kinases, which are known as checkpoint kinases. So they can stop the cell cycle, make an arrest, and either call for the cell to go through apoptosis or have the cell try to repair its damaged DNA and complete to be alive. So if the DNA damage is not repaired, then the apoptosis signal will be the answer to that problem, to the non-responsive DNA damage. So we found out that glycogen synthase kinase 3 beta was inhibited and the cyclin-dependent kinases were overexpressed. More is being done about that to go more into the vivo studies. So this is in the pipeline. Another research was about trying to find simple constructed compounds based on small molecules that are easy to make at the lab but still can be effective. So we tested what we call naphthyl pyruvate derivatives as small molecule kinase inhibitors. Here is an overlay of how they can match the structure of the ATP in binding to its hinge region. And those are the structures of the compounds that we designed. So as you see, they are simple to make, and the nice thing, we were following one of the papers of Dr. Ashraf Abadi, who is not doing anti-cancer, but we decided we would try them as anti-cancer, and they worked. And it's a one-pot reaction. What does it mean, one-pot reaction? You put everything in there, cook it, and come out with your compounds, nice and clean. So we have to think about economy as well, as Dr. Mazar was highlighting. When we did the in vitro toxicity, we had, from these, we had 70 compounds made, so I'm just showing a sample, not to waste much of the time. We had them tested against 3 types of breast cancer and 2 types of prostate cancer cell lines. 3 types of breast cancers that were tested are MCF7, triple positive, MDA, MD435, triple negative, and PT549. And they were found to be more active, the most active, against MCF7 and the prostate cancer cell line DU145. For that, we also parallelly, or on a parallel track, tested them against kinases, VEGFR2 and EGFR. So we were kind of, why not? Let's try them. And they were potent against both of them in a micromolar range again. Okay, so those compounds are active as anti-cancer by virtue of their kinase inhibitory activity. Lucky me. Okay, so let's go a step further. Do they induce apoptosis as well, or what kind of activity other than just the kinase inhibition? So they were found to be inductive to apoptosis on a dose-dependent manner. The best response was seen in MCF7. Cell cycle progression was also tested using a technique known as flow cytometry, and those compounds were seen to be able to arrest MCF7 in the G1 phase and to arrest MCF7 P53 knockdown and MDA MB468 in the S phase. Again, they are populating the cells in a way so that they will not go further. And these are the numbers. Apoptotic markers were also determined. So we determined PARP, PAX, BCL2, caspase 8, 9, 3, which are markers of apoptosis, and we found that apoptosis again was one of the things that these compounds can do. So those are a few of two examples of a few other projects that we are running now to find compounds that are active against kinases. One of the projects that we're working on, again, is not as kinase as anti-cancer, but kinase as anti-Alzheimer, which is also a valid target. By this, I don't want to take further time. I will come to the acknowledgement point and thank my research group in the Zone City, thank all my master's students and PhD students who have worked and collaborated throughout this path of drug design and discovery adventure. Funding agencies بوسول BDSRC, Academic البحث العلمي, and thank very much our collaborators without whom this couldn't have been possible, from Cairo University, Georgia State University in the US, MSA University, Cairo Lomayo University in France, جامعة الملك سعود, جامعة الملك فيصل in case A, and German University in Cairo, Dr. Ashraf is our collaborator from there. And finally, thank you for your attention, time, and
I'd like to thank the organization committee and Professor Ashraf for inviting me in such a conference. My talk today will be on the design, synthesis, and biological evaluation of tamoxifen rigid and flexible analogues for the treatment of breast cancer. I'll start with first the outline of my presentation will be a brief introduction about breast cancer, followed by the research objectives, synthetic schemes adopted in this work, results and discussion, and wrapping up with the conclusion and future prospects. Starting with the introduction, breast cancer is one of the most common cancers worldwide. According to Global Cane, breast cancer is the most common cancer in women, accounting for 25.1% of all cancer cases. And around 1.5 million new cases of breast cancer were identified. Around 500,000 cases of death worldwide were due to breast cancer. So what are the strategies implemented for the management of breast cancer? Management of breast cancer include surgical interference, radiation therapy, and finally systemic therapy. Surgical interference can be done through lumpectomy, mastectomy, and modified radical mastectomy. Also, systemic therapy includes chemotherapy, targeted drug therapy, and hormonal therapy. Breast cancer is a hormone-dependent tumor, and estrogen is known to play a major role in the development and the progression of the disease. Very recent studies have highlighted a possible correlation between serotonin and cancer. Focusing on first estrogen, estrogen receptors are members of nuclear receptor superfamily, known as ER-alpha and ER-beta. Both receptors, ER-alpha and ER-beta, are encoded by unique genes composed of 5 functional domains. Starting with the first domain, which is the A and B domain, it contains activation function number 1, which is a ligand-independent transcription activator part of the receptor. The following domain is the C domain, which is the DNA binding domain. It binds to the estrogen response elements on the target genes. Followed by the D domain, which is a flexible or hinge region. It's the region between the C domain and the E domain. The E domain is the ligand binding domain. From its name, it contains activation function number 2, which is a ligand-dependent transcription activator part of the receptor. And finally, the F domain is located towards the C-terminal domain, which contains cofactor recruitment sites. Over the past years, several hormonal therapies have been developed, such as aromatase inhibitors, selective estrogen receptor downregulators, luteinizing hormone releasing hormone analogues, and finally selective estrogen receptor modulators. Starting with selective estrogen receptor downregulators, fulvestrant is a first-class SERD. It competitively inhibits the binding of estradiol to estrogen receptor, leading to impaired dimerization and degradation of the estrogen receptor, leading to decrease in the transcription of estrogen-regulated genes. The second type are the LHRH analogues. LHRH hormone is a linear decapeptide released from the hypothalamus. It acts on the LHRH receptors on the pituitary gland, leading to the release of the FSH and LH hormones, which stimulates the ovaries and testes for the secretion of estrogen and testosterone. initial increase in the FSH, LH, estrogen, and testosterone leads to desensitization and deregulation of the LH-RH receptors, which consequently decrease the levels of estrogen and testosterone release. And finally, the selective estrogen receptor modulators, which we will focus on today, they can act as both agonist and antagonist according to the type of tissue they act on. SERMs, when they bind to the ER on the uterine tissues, they lead to ER activation, leading to an agonistic activity and increased cell proliferation. On the other hand, when SERM binds to the estrogen receptor on the breast tissues, it leads to ER inactivation, leading to an antagonistic action and decreased cell proliferation. This dual activity can be explained by three reasons. The first one is the differential ER alpha and ER beta expression. The second one is the ratio of co-activator to co-repressor proteins in different cell types. And finally, the conformation of the estrogen receptor induced by drug binding. SERMs are chemically classified into benzothiopenes, indoles, naphthalenes, benzopyrans, and most importantly, triphenylethylines. One of the most important triphenylethylines known is tamoxifen. Tamoxifen, it was approved by the US Food and Drug Administration, and it became the gold standard therapy in the treatment of breast cancer, especially in ER-positive cells. Tamoxifen is a prodrug that needs to be extensively metabolized to yield its pharmacological action. Tamoxifen can be hydroxylated to 4-hydroxytamoxifen by CYP2D6. It can also be metabolized by CYP3A4 to yield N-desmethyl-tamoxifen. Both metabolites will be further metabolized to the more active metabolite, which is endoxifen. 4-hydroxytamoxifen has higher affinity on estrogen receptor more than tamoxifen. Also, 4-hydroxytamoxifen and endoxifen has higher anti-cancer activity on MCF7 breast cancer cells. CYP2D6 is the leading enzyme responsible for endoxifen production. CYP2D6 exists in several polymorphs, and due to this phenomenon, the clinical outcome of tamoxifen differs from one patient to another. Natural genetic variation in the alleles of CYP2D6 lead to several CYP2D6 phenotypes, which lead to the production of less or more active enzymes. It helps us classify our patients into poor metabolizers, ultra-rapid metabolizers, intermediate metabolizers, or extensive metabolizers. Moving on to the second hormone which may be affecting breast cancer, serotonin. What are the physiological functions of serotonin? Serotonin plays a role in heart development, platelet aggregation, modulating intestinal motility, and regulating the vascular tone. There are different serotonin receptor subtypes, ranging from serotonin receptor subtype 1 up to subtype 7. All of them, except serotonin subtype 3, are G protein coupled receptors, and serotonin subtype 3 is a ligand-gated ion channel. So, moving on to the relation between estrogen and serotonin. High levels of 17-beta-estradiol will lead to increase in the levels of tryptophan hydroxylase, which increases the levels of serotonin in our body. It can also decrease the levels of monoamine oxidase enzyme, which leads to also an increase in the serotonin levels. It can inhibit the serotonin reuptake inhibitor transporter, which increases the residence time of serotonin. And finally, 17-beta-estradiol in the presence of progesterone, it increases the levels of ER-beta receptors in our body, which upregulates serotonin receptor subtype A. Very recent studies have shown a possible correlation between serotonin and cancer. What happens when serotonin increases in our body? Serotonin can stimulate mitogenic activated kinases, such as seed-John N-terminal kinase and extracellular regulated kinases. It can also stimulate cortisol secretion, especially in patients suffering from Cushing syndrome, which leads to increase in the levels of growth hormones and factors, leading to increase in the tumor growth. It also mediates proliferation through crosstalk with receptor tyrosine kinase. And finally, serotonin and vascular endothelial growth factor can stimulate a set of identical kinases. Serotonin, which is secreted from platelets, can interact with several neighboring cells, leading to increase in the tumor growth through angiogenesis. After finding a possible correlation between serotonin and estradiol, it may be anticipated that tamoxifen that blocks the action of estrogen may be used as future ligands for the treatment of breast cancer. main research objective was the development of tamoxifen, novel synthesized compounds which which avoid CYP2D6 polymorphism and metabolized by other metabolic pathway other than CYP2D6. New tamoxifen analogues with altered activation pathway via esterases rather than CYP2D6. Also, evaluating the binding affinity on different serotonin receptor subtypes, and evaluating the growth inhibitory effect of the synthesized compounds on 60 cancer cell lines by the National Cancer Institute, including MCF7 breast cancer cells. The structural modifications implemented on the structure of tamoxifen was a chloro group was introduced at the para position of ring A. Also, a methyl group was introduced instead of the ethyl group of tamoxifen. Two series were implemented, the flexible and the rigid series. Flexibility was endorsed through the introduction of a benzylic methylene moiety. Also, introduction of different alkyl and alicyclic amines at the para position of ring B. And finally, introduction of different alicyclic and aryl derivatives, ester derivatives at the para position of ring C. Skipping the synthetic pathways and moving on to the results and discussion. Starting with the structural elucidation, our novel compounds were all confirmed using LCMS, proton NMR, and carbon-13 NMR. Purity via LCMS was obtained more than 90%, and also our compounds revealed E and Z mixtures. The next step was to biologically evaluate our compounds on serotonin receptors. Binding affinity to different serotonin receptor subtypes was done first using primary irregular binding assay to determine the percentage binding affinity at 10 micromolar. Compounds having more than 50% binding affinity were escalated for a secondary screening to determine their Ki value. 11 novel synthesized compounds were tested on 12 different serotonin receptor subtypes. 5 out of 11 compounds showed binding affinity greater than 50%. 3 of them have shown selectivity towards serotonin receptor subtype 2A and 2C. And 3 of them showed selectivity towards serotonin receptor subtype 2A, 2B, and 2C. Compound 9, 13, and 26, all of them showed selectivity towards serotonin receptor subtype 2A and 2C. All of them share a common feature of having an aromatic ester derivative in their group, which may indicate that the aromaticity of the ester groups is important for pi-pi stacking interactions, and these two receptor subtypes may favor lipophilic interactions. Also, serotonin receptor subtype 2A and 2C can tolerate both either a benzyl or phenyl ester derivative. Compounds 5, 6, and 7, all of them share a common feature of having a morpholine ring in the para position of ring B. They showed binding affinity less than 50%. This may be attributed to the presence of the morpholine ring and the oxygen in its group. The presence of the oxygen may lead to increased hydrophilicity, which decreased in the lipophilic interactions of our compounds to serotonin receptor subtype 2A and 2, all serotonin receptor subtypes, not only 2A and 2. 5 of our compounds were selected for secondary screening to determine their Ki values. 4 of them showed Ki values less than that of tamoxifen, especially on serotonin receptor subtype 2A and 2C. And 3 of our compounds have shown, sorry, there's a problem in the animation. 3 of our compounds have shown remarkable Ki values of less than 5 nanomolar, compound 9, 13, and 9, 12, and 13. They showed Ki values of less than 5 nanomolar on serotonin receptor subtype 2C. Our hit compounds, compound 9, 12, and 13, showed Ki values of 2.7, 3.9, and 4 respectively on serotonin receptor subtype C, in comparison with tamoxifen having Ki value of 318 nanomolar, indicating that the structural modifications implemented on the structure of tamoxifen has successfully shown remarkable Ki values. All of them also share a common feature of having an aromatic ester group, which may help us conclude the importance of the possible pi-pi stacking interactions on serotonin receptor subtype 2C. Also, serotonin receptor subtype 2C can accommodate different alkyloxy amino side chains, either propoxy, ethoxy, cyclized, or non-cyclized. Comparing compound 13 with compound 12, they showed Ki value almost the same of 3.9 nanomolar. It may help us conclude that serotonin receptor subtype 2C can accommodate either a phenyl or benzyl ester derivative. Comparing them with compound 9, the only difference between 12 and 13 and compound 11 is the conversion of the aromatic ester group into a cyclohexanoyl ester group. As we can see clearly, the Ki of the cyclohexanoyl ester group or compound 11 had demolished the Ki value of 10 micromolar, which may also help us conclude the importance of the aromatic ester derivative and their interaction with serotonin receptor subtype 2C. Comparing compound 26 with compound 24, moving on to the flexible series, comparing compound 26 with compound 24, also the presence of the aromatic ester group have shown better Ki values of 191 nanomolar in comparison with compound 24 having 1557 nanomolar. Moving on to serotonin subtype 2A, as we can see, comparing compound 26 with compound 13, compound 26 showed better Ki value than compound 13, indicating that the endorsed flexibility may have better Ki values on serotonin receptor subtype 2A. But also we can clearly see that serotonin receptor subtype A can accommodate both rigid or flexible series. Moving on to the anti-cancer activity, which was done by the National Cancer Institute through primary screening to determine the percentage growth inhibition at 10 micromolar. For the NCI results, 6 of our compounds were tested for their anti-cancer activity and for their binding inhibition on different serotonin receptor types. The compounds showed variable growth inhibitory percent, with compound 9 having the highest mean percentage growth inhibition of 81.35% on 60 cancer cell lines and 96.22 on MCF7 breast cancer cell lines. Comparing compound 9 with compound 12, compound 9 bearing an azipan ring, azipan ethoxy side chain, showed almost 2-fold higher mean percentage growth inhibition than the dimethyl aminopropyloxy side chain, indicating that the azipan ring may have a higher lipophilicity and may do better interaction with the 60 cancer cell lines and the MCF7 breast cancer cells. Strikingly, compound 9 having the highest mean percentage growth inhibition on 60 cancer cell lines and the highest mean percentage growth inhibition on MCF7 cell line showed also the highest Ki on serotonin subtype C of 2.7 nanomolar among all our tested compounds. Attempts to find correlation between negative log Ki and the percentage growth inhibition on 60 cell lines have shown a correlation coefficient of 0.604, and also correlation between the percentage growth inhibition on MCF7 cell line and negative log, the Ki have shown a correlation coefficient of 0.814. This indicates, and for the first time, that the anti-cancer activity mediated of this class of compounds, particularly on ER-positive cells, may be mediated via serotonergic receptors. Wrapping up with the conclusion, 24 novel tamoxifen analogues were synthesized. All compounds were tested for their binding affinity and Ki on different serotonin receptor subtypes using primary and radioligand binding assays. The 3 most potent compounds are compound 9, 12, and 13, which belongs to the rigid series. They showed Ki values lower than that of tamoxifen by 118, 82, and 80 folds respectively. The most important structural features for triphenyl ethylene scaffold as serotonin ligands are an aromatic moiety attached on ring C and the basic amine either cyclic, alicyclic, propoxy, or ethoxy side chain, and also the effect of different spacers should be tested. Our future prospects: further metabolic studies in the pharmacokinetic profile are to be performed to determine the exact rate of metabolism of the novel compounds, planning for in vivo testing for the most potent compounds of hydroxylated and estrified series, and compounds are currently screened for the functional assay to determine agonistic-antagonistic action on serotonin receptor, as anti-estrogenic activity and screened for inhibition on 60 cell lines. Thank you. I'd like to thank Professor Ashraf Abedi for his main supervision on this thesis. Thank you so much, Dr. Ashraf, for supervising me and for giving me such an honor to be in the Abedi Research Group. I'd like to also thank my co-supervisor, Dr. Nermeen Salah, for her endless support. I'd like to thank the STTF for funding this project, and I'd like to thank the National Cancer Institute and Professor Brian Roth for performing the biological assay. Thank you.
Thank you.
Thank you, Dr. Ashraf, for inviting me to this conference and to give a short talk about my research work.
باشا هي كان مطلع روح شوف بقى عشان تخش جوه.
To give a short talk about my research work that involved the synthesis of novel aryl physostigmine carboxylic acid amide derivatives as cytotoxic agents against hormone-dependent carcinoma cell lines. In this short presentation, I will give a short introduction, followed by the research objective of my work, then the synthetic routes followed to design compounds. So again, I'll talk about the synthesis of novel aryl physostigmine carboxylic acid amide derivatives as cytotoxic agents. I have to move these slides.
cancer, the complexity of cancer as a disease can be reduced into 6 main hallmarks that govern the transformation of a normal cell into a cancerous or malignant cells. These 6 main hallmarks are: the cancer cell has self-sustained growth signaling, has insensitivity to anti-growth signals, the cancer cells can evade apoptosis, and has limitless replicated potential. The cancer cell has the ability of sustained angiogenesis and has the ability of tissue invasion and metastasis. As my colleague Myrna discussed the statistics of cancer cases worldwide, lung and prostate cancer represent the most commonly diagnosed cancers among males according to the most recent 2012 statistics, and among females, breast cancer represents the most commonly diagnosed cancer among females. GPCRs represent a superfamily of cell surface membrane signaling proteins, and they contribute to multiple physiological functions in addition to multiple diseases including cancer development and cancer metastasis. That's why they represent a key target for drug discovery in cancer development and cancer prevention. As we all know the structure of GPCRs, there are 7 transmembrane domains coupled to a G protein. The G protein is a trimer of alpha, beta, and gamma subunits. When an agonist binds the GPCR, this causes a conformational change in the receptor that is transmitted to the bound G protein. The G alpha subunit, when undergo transformational change, it simply exchanges GTP in replacement for GDP. When an agonist binds GPCR, this causes a conformational change in the receptor. This conformational change causes dissociation.
بس تمام. ان شاء الله خير، ان شاء الله باذن الله. سلمى
This causes the G alpha subunit to exchange GTP in replacement for GDP. And now the activated G alpha subunit dissociates from the beta gamma dimer and from the receptor. And now the dissociated alpha subunits and beta gamma dimers interact with other intracellular proteins to continue the signal transduction pathways that leads finally to various cellular responses, including proliferation, survival, migration, angiogenesis. And of course, these cellular responses contribute to tumor growth and metastasis. Among the agonists for GPCRs are LPA that we will focus on our talk today. LPA is lysophosphatidic acid, and it is a simple phospholipid composed of glycerol skeleton, composed of glycerol backbone and phosphate group and 1 fatty acid. LPA has many physiological and pathological effects, and these effects are actually mediated through a series of GPCR receptors. At least 6 identified LPA receptors have been identified in terms of LPA1 to LPA6. Autotaxin is the main enzyme responsible for the production of LPA in our bodies, and it's simply lysophospholipase D, so it removes the choline group from LPC, lysophospholipid choline, to produce LPA. Many research studies showed that LPA has a major role in cancer cell proliferation, migration, metastasis, and resistance to therapy. And actually, an elevated level of LPA and/or elevated or overexpression of LPA receptors have been reported in many cancer types, including breast cancer, ovarian cancer, prostate cancer, melanoma, and hepatocellular carcinoma. And actually, this guides us to the research objective of our work. The simple structure of LPA plus its major effects on cancer cells led the researchers to develop therapeutic agents based on the LPA scaffold. So the main series of compounds were designed in 2005 by the Door Research Group, and they were the serenamide phosphate. These compounds show high cytotoxic activity in the low micromolar range, but they have reduced selectivity. So further modification was done to improve the selectivity. A new series of compounds were designed, which are the 2-aryl-4-oxocyazidine amide. These compounds were of improved selectivity, but of reduced potency. So further modification was done to produce a new series of compounds, which are the 2-aryl-thiazidine 4-carboxylic acid amide, ATCAAs. These compounds were of higher potency and improved selectivity compared to the serenamide phosphates. And actually, high cytotoxic activity of these compounds was reported against prostate cancer cells and skin cancer cells. So, the aim of our work is to design novel aryl-thiazidine carboxylic acid amide derivatives and exploring their cytotoxic activity against the previously studied prostate cancer cell line PC3, and for the first time to explore their cytotoxic activity against breast cancer cell line PC3, breast cancer cell line MDA, MB231, and the ovarian cancer cell line SKOV3. Further structural modifications were adopted. First, in the 2-aryl part, we tried fluorinated phenides, fluoromethoxyphenides, and hydroxymethoxyphenides. And for the amide side chain, we tried hexyl, dodecyl, and octadecyl amide derivatives. We also tried masking the secondary amine of the thiazidine nitrogen with Bock group to explore if it is essential for cytotoxic activity or not. These are the synthetic routes followed to design our compounds, and all the compounds were analyzed using LCMS, proton NMR, carbon-13 NMR for structural elucidation. Actually, the results of our work were very interesting. As we mentioned earlier, we tested the in vitro cytotoxic activity against three main cancer cell lines, PC3, SKOV3, and MDA-MB231. This table shows the biological activity data. Actually, we designed 30 compounds. 17 out of the 30 compounds were able to inhibit the proliferation of cancer cell lines at a screening dose of 50 micromolar. And the IC50 values were determined for compounds showing more than 50% inhibition and 50 micromolar dose. And now for the structure-activity relationship studies for the compounds regarding the effect of the amide side chain length, it was found the dodecyl derivatives were more potent than the octadecyl derivatives, and the hexyl derivatives were the least potent, as shown by the IC50 values for the corresponding compound. It was also shown that the activity of the dodecyl derivatives was maintained regardless of the pattern of substitution on the 2-aryl part. For example, the activity was maintained when the 2-aryl part has an electron withdrawing fluorine atom or an electron donating group like methoxy group. However, the activity of the C18 derivatives was markedly changed by the pattern, and type of the substitution on the 2RI part. And now, when we discuss the structure-activity relationship regarding the 2RI part, when we try the fluorinated phenyl, generally the parafluorophenyls were twofold higher potency than the metafluorophenyls. And the orthofluorophenyls were the least potent. But when we introduced a second fluorine atom, this led to a marked decrease in the cytotoxic activity. This could be due to the increased electron withdrawing effect by the two fluorine atoms on the phenyl ring. When we added a methoxy group to the fluorophenyl substitution on the 2RI part, we found that there is a marked increase in the potency due to the electron donating effect of the methoxy group, as shown here in the dodecyl derivative. And this boosting of activity was also shown in the octadecyl derivative. Actually, compound 28 is one of the best compounds, as it shows submicromolar IC50 against the 3 cancer cell lines, as shown here from the IC50 values. A more pronounced boosting of potency by the 4-methoxy group was observed with the C18 derivative. And when we introduced 2 electron donating groups, hydroxymethoxy phenyl, when compared to fluoromethoxy phenyl, this led to more increase of cytotoxic activity. As shown here, actually compound 25 showed an IC50 value of less than 0.1 micromolar against the MDA and B231 cell line. And regarding the effect of bulk protection, it was found that the masking the secondary amine of the thiazidine led to a remarkable diminishing in the cytotoxic activity. So the free nitrogen is found to be essential for cytotoxic activity. Actually, this graph shows the preferential activity of the compounds towards the 3 tested cancer cell lines. And we can conclude that the MDA and B231 cell line was shown to be the most sensitive to the synthesized compound. And this is very important finding, as we know that the MDA and B231 is a triple negative cancer cell line, and it is an aggressive type of breast cancer. So this class of compounds will provide a promising ideas for developing effective treatment for this triple negative cancer type. So in conclusion, compounds were designed. Many of them showed submicromolar potency against 3 cancer cell lines, and the MDA cell line was found to be the most sensitive. And the structure activity shown that the dodecyl derivatives were more potent than the octadecyls and the hexyl derivatives. And the potency is increased when electron donating groups are from the 2-phenyl system, and the secondary amine of the thiazidine ring is essential for activity. And for future remarks, the safety and the selectivity of the promising compounds are to be investigated, and the future efforts are to be done for the synthesis and evaluation of pure individual stereoisomers, and in vitro and in vivo studies are to be done for the compounds, and of course further biological studies are required to know the exact mechanism of action of these compounds, and planning for testing the cytotoxic activity against different cancer cell line types. Finally, I want to thank Professor Dr. Ashraf Abadi for being the supervisor for this research work, and Dr. Muhammad Abdelali Abdelhanim for being the co-supervisor, and for giving me the opportunity to be among the research group. And I want to thank Professor Gary Piazza for running all the biological activity data in Nature Cancer Institute, University of Alabama, USA.
Thank you, Eamon. Any questions?
No, fix it.
Doubling time. Doubling time.
Is the doubling time?
Doubling time. Cell line doubling. Cycle time. Time needed for one division. This is the most sensitive one. The sensitivity could be due to the difference in doubling time, so it needs more time to reach one of them, or really due to the treatment. So
Various factors could influence the selectivity profile of the compound. So in the future prospects, we showed that we need to do further studies for selectivity.
No, my simple question is doubling time, same or not? For the cell line, same?
It's all 48 hours.
Thank you.
Can I ask a question? Your compounds having two chiral centers. Okay, so did you measure the optical activity of this compound? I know you are working on racemic optically impure compound. So
No, we didn't measure the optical activity, but we did with the compound is racemic maximum because we haven't been able to spray it using the iron.
okay, thank you. It's difficult.
This question of characters really if you really targeting drugs to work on receptors, so the enantiomer will be more. You will have in your cell at least one of them active and the other will be less.
Actually, this is not a de novo work. We already worked on previously studied the compound, the same series, and all the previous work worked on racemic mixtures. Until now, neither we nor they were able to separate the isomers. And actually, we said it in the future prospect.
Is it possible to specifically synthesize one enantiomer? Not to spread it because we tried that, where we've been successful with valproic acid derivatives. And substitution in one chain of the valproic acid like entire center. Always, it was not easy, of course, with the German, my German colleague there, he was able to sensitize till one, you have a really pure and we found big differences between
We'll try to make this maybe later, but so far we're working with this mixture of isomers. But for asymmetric senses, this is not an easy task for us at least. I have actually a comment regarding Professor Mazar talk. It is very interesting talk. And if I were in the Egyptian Pharmaceutical Authority, I would like to adopt this technique of repurposing of existing drugs for different uses, because it will save us as a developing country a lot of efforts, money, and of course will make us more certain about the outcome. And I would like here to highlight that this is not even a tactic for developing countries, it is for developing countries as well. So if you look for example, you said, it was first introduced for treatment of angina and hypertension, and then repurposed for erectile dysfunction. Now even Viagra is repurposed for pulmonary arterial hypertension. So this is an example. If you look to Novartis, if you look to isotretinoin, it is the same type of constituent in Sandomigran for treatment of migraine, and in Mosigo, which is an appetizer. So it's a dual use. Also among the emerging repurposing, we have chlorcyclizine, which is mainly used an all-time antihistamine. It is now officially repurposed for treatment of different genotypes of hepatitis C virus. So this may be also a way to reduce the cost of treating of hepatitis C in Egypt. Tamoxifen and torimifene, which are our compounds we are interested in, even approved by the FDA for treatment of Ebola virus. So actually there are a lot of chances for Egypt, yes, in order to have treatment for diseases which are important for us by existing medications. And I think in this case we don't need license and we don't need a lot of registration process or legislations. No, they are present in the market, and I think this may be an Egyptian chance to develop at least.
2 points.
Yes.
Actually, registration, you have to register your application. This is different than, I was surprised to know that in the Egyptian patent office, they would not patent a drug for a new indication. You can do that in America, but not in Egypt.
Actually, in Egypt, you can patent the molecule, but not patent the use. This is the Egyptian Patent Office. Yes, I know the rule about this. I was a member of the Egyptian Committee for Intellectual Property. And yes, in Egypt we don't patent the use, we patent the molecule. Yeah, and again also you raise the point of targeted medications by liposomal and nanoformulations, and I think this is also among
This is a good
very,
prospect.
yes.
You can overcome, if you have a drug, simply they started to use this, prolonged effect, delayed effect, because the ones which are rapidly metabolized or rapidly clear, you can put it in a formulation so that they sustain effect. But now, with these liposomes, you can have new attachment to the outer layer to target, say, liver. So it will go, if you give a dose, which is instead of exposing the whole body to a huge amount of the drug and mostly produce toxicity, you can have a smaller molecule, but targeting to one organ, or much better to a cancer cells. So you're building up a huge amount of concentration there, relatively minute amounts in this circulation. I think this is a very promising approach.
One last point you raised, which is also suitable in Egypt, which is drug combinations.
Yes.
And I think also this point raised in Dr. Reem's talk, if you have drugs attacking different checkpoints in a particular pathological conditions, so they have different pharmacodynamics, but of course they should have similar pharmacokinetics. So this is the golden
But
rule.
it had not the rule. You have to test it, you have to prove
Yes,
it.
you have to prove, yes,
You have
but
to prove it.
in
So
order
it
to
can
be,
be customized, some customized clinical trial.
Yeah,
It
so-
would be faster.
Yeah,
You
yeah.
have to start from there.
so usually they are looking for drugs with different mechanisms of action, but their pharmacokinetics should be similar so that they are existing all the time in blood altogether. So this is
The basis of
yeah, this is the basis. Okay. So other questions? Remarks? Like by this, I would like to thank you all for your time and good listening, and inshallah نشوفكم في next year inshallah or next conference. Thank you.